Hello, my name is Benjamin Miss, and this is Introduction to Cognition. Today, we're going to be talking about neuroscience. Neuroscience and cognition. The mind is inexorably linked to your brain, which means to fully understand cognition, we need to understand the organic machine responsible for your thinking. Your brain uses approximately 20% of your body's energy, and 15% is expended in signaling or processes involved in neuron communication. The brain is made up of nearly 100 billion neurons, which may each form connections with up to 10,000 other neurons. In addition, your brain has approximately 100 billion glial cells, which are thought of often as support cells, but many of them are instrumental in neural communication. The brain is highly efficient. In 2013, a team at the Okinawa Institute of Technology Graduate University in Japan successfully modeled 1 second of processing in an artificial neural network with fewer than 2 billion neurons. It required over 80,000 processors, used a petabyte of system memory, a petabyte is 100 terabytes, and took 40 minutes. Someday, machines may be able to model what we do. That day is not near. The human brain can perform nearly 1,000,000 times more calculations than your home computer and uses up to 10 times less energy. We are living in a golden age of neuroscience. Historically, change was slow, and it is only with technology developed since the 1970s that we can look with accuracy inside the brains of living humans. From the ancients who believed that the seat of the soul was the heart, to 18th century anatomy textbooks, much of the work on the brain was guess and conjecture. There are stories of trephination, or drilling holes in the head to remove skull shards after fracture, relieve migraines, and cure epilepsy. It was used for treating mental illness, and stories suggest the holes were part of the ancient supernatural explanation for mental illness, and the holes allowed for the demons to escape. The holes were generally large, the size of a $50, and most patients appear to have survived, as evidenced by the healing of the skull. Trephination is still reported to be used today for medical treatment by those in less developed areas, and others for pseudoscientific reasons such as enhanced consciousness. However, over that time period, little changed in our understanding the brain. Trephination was not brain surgery, as they didn't go past the 3 layers of meninges that protect the brain. It may have helped in some cases, especially to relieve pressure on the brain, but it did not show any great understanding of the neural tissue that resides inside the skull. By the 18th century, anatomists knew nearly as much about the general structure of the brain as we do today. The function of the brain was so misunderstood that Franz Gall proposed phrenology as a method for understanding behavior. Gall had an interesting idea, which was that our intelligence, personality, and behavior are influenced by the brain. He believed different areas of the brain did different things. So people who were particularly intelligent in an area would share increased brain area, and people with criminal behavior would also share brain features. For any shared trait, people should have similar brain function. This was not a terrible idea. There are many who support the modular view of the brain, which is that the brain areas are organized for various different functions. However, Gall took his idea too far and used cranioscopy to measure the shape of the skull. He looked for similarities in groups and, of course, found them. This is an example of small sample size. If I take 5 students in a psychology class and 5 students in an anatomy class, I will find some commonality in the skull shape of the psychology students and some commonality in the skull shape of the anatomy students. However, that would not convince you. Skull shape determines whether you are in a psychology or anatomy class. That would be ridiculous. Yet in Gall's time, people readily believed that skull shape would explain personality. Instead, it was the story of Phineas Gage and the patients of Paul Broca, who was influenced by Gall, and Carl Wernicke in the mid-19th century that began to show some understanding of the function of the brain itself. Gage was a railroad worker and had a long metal rod blasted through his brain, damaging his frontal lobe. Widespread attention in popular media highlighted the mental changes that accompanied his injury. The study of brain-injured patients still goes on today. It was the first real way to understand the brain. For Broca and Wernicke, they had to study the behavioral changes in their patients and then wait until the patient died in order to examine the damage to their brains. Patients who had strokes would often have clear areas where the brain was destroyed, suggesting that if a patient lost an ability, that part of the brain was responsible. The study of neuroscience is generally focused on biology and chemistry, but part of the work concerns the link between brain and behavior, and it will be addressed here as a link between the brain and the mind. The brain is a highly efficient computing machine. If we can understand the function of the brain and how it relates to our experiences, we can solve Chalmers' easy problem of consciousness, which relates to what is happening in the brain when we're having an experience. Modern neuroscience has already given us a great deal of information into how various structures and networks in the brain contribute to our conscious perceptions. The building blocks of the brain. The brain is made up of neurons and glia. We generally focus on neurons and ignore glia, but without glia, your brain could not function. Some glia, known as astrocytes, are intricately involved in neural communication, regulating the flow of neurotransmitters. Neurons communicate in 2 ways. One, through electrical impulses using charged ions, and the other through chemicals known as neurotransmitters. Neurons come in many different forms, but the structures that make them different from other cells are the dendrites, which generally receive information from other neurons, and axons, which send information to other neurons. Between axons and dendrites is the tiny space called the synapse. Neurotransmitters are released by the axon terminals and diffuse across the synapse to where they attach to receptors on dendrites. The most well-known neurotransmitters include serotonin and dopamine. The most common ones are glutamate and GABA, which are both amino acids. Glutamate is the brain's primary excitatory neurotransmitter, and GABA is the brain's primary inhibitory neurotransmitter. This tells us that sometimes a neuron will tell another neuron to fire, and sometimes a neuron will tell another neuron to not fire. Many neurons in the brain fire spontaneously, and the amount of neurotransmitters they receive tell those neurons to speed up or slow down. Some neurotransmitters, like glutamate and GABA, will primarily send a signal that has an immediate effect on firing rate. However, many synapses can also allow for the neurons themselves to change. A neurotransmitter can send a signal that causes long-term change into the neuron that might strengthen or weaken the synapse. If an adult humans do produce new neurons, it is rare. Therefore, in order for us to learn and store information, neurons must be able to change. Some ways neurons can change are by adding or removing synapses, changing the balance of neurotransmitters, or adding or deleting receptors for those neurotransmitters. The effects of neurotransmitters are still not completely understood, and some are difficult to measure in a living brain. Medications given to treat depression, such as Prozac, can start influencing the effects of serotonin right away, but can still take weeks before the patient feels better. This tells us that the neurological cause of depression is not fully understood. In fact, the pop science link between serotonin and mood is based on the fact that drugs that increase serotonin availability can lessen symptoms in depression. Other drugs used to treat depression affect other neurotransmitters in addition to serotonin. And recent research shows that Prozac is one of the least effective depression treatments, but one of the most well known, giving us more evidence that pop science in advertising must be carefully investigated. Just like prescription drugs that alter the mind, street drugs that people take for the high also affect neurotransmitters. The most common neurotransmitters involved is dopamine. Drugs that alt-boost dopamine contribute to a feeling of euphoria or well-being and tend to be highly addictive. Some hallucinogenic drugs, like LSD, work in other ways that can distort reality. For instance, some drugs will affect some types of neurotransmitter receptors, but not others. So dopamine may be increased in some brain areas, but not others. Time perception is likely related to some types of dopamine, norepinephrine, and serotonin receptors. Brain anatomy. The four main lobes of the brain are the frontal, parietal, temporal, and occipital. Each lobe has historically been associated with specific function, but modern studies show that in many cases there are wide individual differences. The frontal lobe is associated with movement and planning. The parietal lobe is implicated in our senses and spatial abilities. The temporal lobe processes sound and understanding language, and the occipital lobe is dedicated to vision. While this may oversimplify the brain, it is a good starting place for understanding brain function. If you were to cut the brain in half down the middle, you would find that the two hemispheres resemble each other, but are not perfect mirror images. The two hemispheres are mostly separated and are connected by a thick band of neural tissue called the corpus callosum, sending information back and forth. At the center of the brain is the thalamus, which is like the central station connecting and tying everything together. Around the thalamus, you have the basal ganglia, associated with learning and memory for motor tasks, and below the thalamus is the hypothalamus that regulates our hormones. The amygdala is associated with emotional impulse, and it is right next to the hippocampus that is responsible for our memory of our experiences. This is a good example of why knowing the geography of the brain helps us. It should make logical sense that emotion is tied so tightly to personal memory, as strong emotions help us form positive bonds and also remind us of what dangers to avoid. The brainstem is involved in automatic processes like breathing and arousal, and the cerebellum is associated with movement, learning, and some types of memory. Localization of function. Certain brain areas are traditionally connected with specific complex abilities, though there is a great deal of variability in brain structure between people, particularly in the cortical areas on the surface of the brain that represent areas of higher functioning. Some neuroscientists argue that localization is less important than other factors, such as the amount of cortex involved in complex activities. Still, it is useful to understand some specific areas, even though the function of these areas are not universally accepted. In the James Bond movie Spectre, the villain attempts to drill into Bond's brain and remove his ability to recognize his girlfriend. As is usual with science in movies, there is some truth to this, but it is not quite correct. The villain's drill would likely cause a great deal of damage beyond simply removing the ability to see faces, since he was drilling in the wrong place, and the villain would probably have to drill into both the left and right hemispheres. This area is called the fusiform face area, the FFA, and is present in both hemispheres, though it is usually larger in the right hemisphere. It is located on the bottom surface of the brain in the temporal lobe near the occipital lobe. The existence of the FFA is a testament to our social nature and the importance of fast and accurate facial recognition. The FFA is also involved in discriminating similar objects and is particularly active in individuals who are experts in a specific class of items, such as cars or farm animals. Two more specific areas are associated with language, called Broca's area and Wernicke's area. First investigated in the 1800s and named after the researchers who discovered them, there is debate about the exact location of these areas, but they are generally accepted to represent two different aspects of language. Language is associated with the Sylvian fissure, which is the large indentation between the temporal lobe and the frontal lobe. This should make sense, since the temporal lobe is associated with hearing and the frontal lobe is associated with movement. This general area is involved in understanding and producing speech. Broca's area is in the frontal lobe above the Sylvian fissure and is associated with producing speech. The intricate movements of the mouth and vocal tract require a great deal of motor control. Wernicke's area is in the temporal lobe, just behind the Sylvian fissure. It is associated with understanding language. For a deeper look at the various brain areas, it is worth considering the work of Corbinian Brodmann, for whom the term Brodmann areas are named. Though not obsolete, describing brain locations as Brodmann areas is not as common as it once was, likely due to the difficulty of memorizing the approximately 50 numbered locations. It is much easier to call Broca's area the inferior frontal gyrus, since inferior means at the bottom, frontal tells us the lobe, and gyrus means raised area. Using descriptive terms allows for a rapid understanding of location, even if we're not talking about a specific structure, but rather a general location. Locating your exact Brodmann area locations would require an individual study of your brain. Therefore, when looking at a CT scan or MRI of a living person, it is easier to describe location using the visual landmarks of the lobes than by Brodmann areas. Brodmann chose his areas based on the tissue, arguing that the layers of the cortex and types and density of cells varied enough to identify neighboring areas as distinct. Understanding that there are clear differences in the brain tissue in different areas lends support for the notion of specialized brain areas. The study of the brain yields information at a rapid rate. Interesting areas of investigation include the recent discoveries of new connections between the brain and immune system, and of a link between neurons in the gut and neurons in the brain. New information is helping to shed light on neurological diseases and processes that contribute to aging in the brain. The search for cures for neurodegenerative diseases, such as Alzheimer's, promise to keep researchers busy for decades. Methods of neuroscience. Electroencephalogram, EEG. In a rather short period of time, we have come to understand a great deal of the function of the brain, including various roles of neurons, glia, and neurotransmitters. Various brain areas do seem to be rather specialized for different things. Much of this knowledge comes from technological developments for investigating the workings of the brain in healthy individuals. The first modern neuroscience technique to develop was EEG, which was introduced in the early 20th century, but required the rapid advancements in computer processing in the mid to late 20th century for us to see the value of the technique. It begins with simply putting electrodes on the scalp to detect the tiny changes in activity in the brain, since muscle activity produces a much higher electrical signal than the brain. It was difficult to read and understand EEG. The good news is if you want to do EEG, the technology has become so inexpensive you can buy a rudimentary setup for a few $100s and perform it with your computer at home. EEG is a measure of overall neural activity. Neurons produce electrical potentials, and the variations in these potentials is measurable. However, much activity is random, and the EEG of an awake and alert person is like a jagged line with small, fast changes. The first major use of EEG was studying sleep. It helped that there was comparably little muscle activity during sleep to interfere with the neural signal. As people fall asleep and into deeper sleep, these waves get larger and larger and slower, suggesting that more neurons are syncing up and firing together rhythmically. This happens repeatedly over the course of 1.5-hour cycles while we sleep. There are two other main ways of using EEG that developed as technology improved. Event-related potentials, or ERPs, involve repeated stimuli and averaging the responses. When a person is awake, there is so much random noise being produced by the brain and scalp muscles that a single stimulus is useless. However, if you take a long series of random numbers from -100 to 100 and average them, you get a number close to 0. If you present the same type of stimulus over and over, and then average the responses, a clear wave will be visible, and the characteristics of that wave will vary depending on the stimulus. Often, presenting two types of stimuli in an experiment, such as faces that are smiling and faces that are frowning, will lead to two different waves to compare once the stimuli are averaged. It can then be demonstrated that the brain is reacting to the two stimuli types differently, even though EEG cannot tell you much about where in the brain the stimuli are being processed. The last main way of using EEG is more recent and developed due to the increase in computing power. A piece of the wave is decomposed into the component parts. It is called steady state. Computerized tomography, CT. CT scans are based on X-rays, but they allow for much more detailed information. For instance, if you want to see an X-ray of a bone, it is easy to do so. For internal tissue, however, you do not receive as much information. A patient is placed in a tube-like machine, and multiple X-rays at different angles are combined in order to provide more internal details than a traditional X-ray. Interestingly, the general machine appearance is similar to PET and MRI scans. Human brain CT scans have been done since the 1970s, allowing for anatomical structures of live patients. Tumors, stroke damage, aneurysms, and other brain problems were able to be identified in living patients and have led to easier and safer treatments. The only downside is that CT scans are X-rays, and many people find it frightening to be exposed to the inherent radiation in this kind of scan. Research suggests that for adults the risk is minimal, and that when there might be bleeding in the brain, a CT scan is likely the best option. CT scans can tell you about the structure, but they cannot give you direct information about function. Positron emission tomography, PET. PET scans were developed during the 1970s and gave the first detailed look at localized brain function in living organisms. When looking at the brain, PET uses radioactive elements usually combined with glucose molecules in order to infer activity. The idea is that when a brain area is performing a specific function, it will have a higher energy need and will use more glucose. Research volunteers would be injected with this radioactive element combined with glucose and then presented with stimuli so researchers could see which brain areas were most involved. PET scanners are extremely expensive to build and use. The radiation dose from the tracer element is about 7 times greater than the radiation exposure for a CT scan, though it is not considered to pose a major increased cancer risk. After the tasks were completed, research participants would be placed in a positron detector. When radioactive elements decay, they emit positrons in opposite directions, which means the detector would be able to get an idea of where those positrons originated, and a visualization of brain activity could be created. The first heat map type color images of brain activity relied on PET technology. The later development of combined CT and PET scanner allowed for even greater precision. Functional magnetic resonance imaging, fMRI. fMRI grew out of an application of standard magnetic resonance imaging. MRI is an imaging technique that is not specific to the brain, though the study of the brain is an important application of the technology. MRI machines are present in hospitals and clinics around the world. These machines use a powerful electromagnet to cause atoms to align with the magnetic field. Though expensive to build and maintain due to supercooling required to keep the magnet functioning, MRI machines have been shown to be worth the expense. Unlike CT scanners, MRI machines do not expose patients to dangerous radiation and can be used repeatedly on the same person. When the magnetic field is applied to a living organism, a portion of that person's hydrogen atoms in water will align, allowing the machine to scan the type and density of various tissues based on the density of water in the tissue. fMRI takes this a step further by taking continuous scans of the brain while the person performs various tasks. fMRI takes extremely rapid scans and, instead of looking at tissue density, looks at the properties of iron atoms in hemoglobin. This allows researchers to see how much oxygen is in the blood, comparing hemoglobin that carries oxygen and hemoglobin that does not. The idea behind fMRI is that when you begin to use an area of your brain more, your body will send more oxygenated blood to that region. This increase in blood oxygenation is then detectable by the fMRI, and we can then see how various regions are involved in processing different stimuli. fMRI, though only in use since the 1990s, is responsible for a massive amount of our knowledge about brain function. Many experiments involve presenting two stimuli that differ by only a single factor. For instance, you can put someone in fMRI, play conversation, and play music. Then while the person is listening to conversation, you scan your brain to see what is active. And when they listen to music, you scan their brain to see what is active. By comparing the two scans, you are able to see differences, which can tell us how these two tasks are similar and how they are different in terms of neural function. Transcranial magnetic stimulation, TMS. TMS is also referred to as rTMS, has been used in research since the 1980s. However, it has taken time to develop the technology. TMS uses a strong magnetic field, similar to the strength of an MRI machine, but focuses the entire field on a small area in the brain. This is useful since most higher level brain activity is near the surface and can be influenced by the technique, which can reach a brain depth of less than 1 centimeter. The benefit of magnetic stimulation is that a magnetic field can pass through the skull, but the field loses strength rapidly. There are two main ways TMS can be used: either disrupt brain activity or enhance brain activity. In research, TMS can be presented with stimuli to disrupt processing of that stimuli by creating what is sometimes referred to as a temporary lesion. A brain lesion is an area of damage, and lesion studies have proven invaluable in understanding functional anatomy. TMS gives researchers the opportunity to create temporary, reversible lesions in healthy adults, which allows the participants to act as their own control group. it is possible to see the before and after effects of stimulation to a particular brain area. The ability to enhance activity has allowed the technology to be used to treat psychiatric conditions, with FDA approval for use in depression as of 2012. Patients receive multiple treatments of TMS applied to their left dorsolateral prefrontal cortex, which is approximately between the edge of the left eye socket and the top of the skull. The procedure has shown remarkable results, especially in patients who have not responded to other treatments for depression. Transcranial direct current stimulation, TDCS, is similar in methodology to TMS, but uses electrical stimulation rather than magnetic stimulation. It is gaining popularity because individuals are able to purchase these stimulators directly, and you do not need a prescription for treatment. However, electrical fields do not pass through bone very well, and research has not shown strong evidence for the efficacy of this technique. Diffusion tensor imaging, DTI. DTI is a special kind of diffusion-weighted imaging that tracks the movement of water molecules in tissue. Since tissue is not uniform, tracking diffusion can show detailed structure in the brain and other organs. One of the first neurological uses was in tracking brain changes after a stroke, since tracking the diffusion of water molecules can show changes in the brain that are not obvious on structural images such as CT and MRI. The structure of neurons allows DTI to track the diffusion in the direction of the neural connections where there are large tracts of axons running in the same direction compared to areas where connections are less uniform, since water will diffuse within the axons at a faster rate. For instance, the corpus callosum is a massive group of neurons that connects the left and right hemispheres of the brain. The idea is that if there are a lot of axons connecting two areas within a hemisphere, these two areas must be working together on important tasks. DTI is clinically valuable in assessing neurodegeneration, the extent of brain damage, and nerve disorders. Newer techniques and the future of neuroscience. In order to truly solve the easy problem of consciousness, we will likely have to be able to observe the complete, real-time function of a living brain. We have seen that this is currently out of the range of our computational capability, though researchers continue to work on advanced models of neurons and brains. Our current resolution with MRI is generally 0.5 cubic millimeters, and increasing the power of the magnet offers only slight increases in resolution and risks patient discomfort. We will probably see a complete model of a functioning brain in our lifetimes, though the goal is still far from our current reach. Quantum computing is promising, yet it often is misunderstood, and it is not clear whether quantum computing is even possible. Some newer areas of investigation include spectrometry and implants. Spectrometry is an application of MRI that goes beyond anatomy, function, and diffusion in order to look at the distribution of molecules in a given sample. While we cannot see the 0.00002 millimeter wide synaptic gap in a living brain, we can attempt to get an idea of neurotransmitter distribution in various parts of the brain. Spectrometry can detect concentrations of molecules like neurotransmitters, allowing us to infer how those neurotransmitters function in various networks in the brain. Neural implants have been used for decades in order to help regulate function in damaged brains. For instance, in some patients with Parkinson's disease, electrodes can be used to stimulate the thalamus and basal ganglia to regulate dopamine production and ease symptoms. Neural implants are also seen as the next step in brain-computer interface. Non-invasive brain-computer interfaces can be designed using EEG and other traditional methods of neuroscience, but invasive methods offer the best promise of rapid, multidirectional information flow. It seems likely that at some point in the coming decades, it will become common for people to electively have electrode arrays implanted in their brains to allow for direct connections to computers. I hope you enjoyed our discussion of neuroscience in relation to cognition today. I look forward to future discussions. Thank you.